So the real title of my talk should be is Big Data, Big Computation, Big Problem, and Big Deals. And really, ENCODE is a monster. It's a huge project. The CRG is a mid-sized partner, and my group is a tiny, tiny partner in this project. But that's the goal. The original goal of ENCODE was very clear. We have all of these things going on in different cells, different tissues. Let us measure everything we can measure, everything that seems to be relevant at a given time. And at the time, this was DNA methylation, open chromatid, RNA binding, RNA sequences, transcriptomics, chipsec. And it's not because these readouts are supposed to be the most informative. We do not know what are the most informative readouts in a cell, but it's because these are the things we can measure, and one has to be humble in this. This effort is driven by next generation sequencing. And so right now, the community is trying to measure everything it can through next generation sequencing. It does not mean that the ultimate answer is in this type of data. It means that this is what we can measure at industrial scale. And so the ENCODE project that is now entering its fourth phase, they are discussing the new grant, was an attempt to characterize all the active regions of the genome in as many conditions as possible. Initially cell types, but in the newer version this has moved to organs. Okay? And that's something that will be familiar to many of you because you in toxicology have typically this kind of semi-military organization, okay, where you have huge data warehouse and everybody has to produce what they have to produce now and with the right format and all these kind of things. And if you know, there is often a little bit of discussion between European and American science, but if there is one thing Americans are really outstanding at, it's really organizing this kind of power warehouse to produce data. And they've been outstanding in these kind of things. And of course you have data production and data analysis centers that are very nicely separated. And one of the very nice legacy of ENCODE is that data has been produced in such a nice way that even though the analysis may vary through time, it will be possible. There is a real legacy that is to stay from ENCODE, I think. So of course you have to throw in numbers. I think that the most striking here is the one below. Now it will be cheaper, but at the time the cost of teleconference was £49,000. You know, so that's a lot of talking. And this is the first ENCODE. This looks tiny like now. We are talking about 15 terabytes of data. We'll be the latest phase. We are in petabytes. Okay? And of course that led to a lot of publication. What is quite important here is these numbers. You get the ENCODE publication and they grow more or less linearly, but you have non-ENCODE publications, the publications that are using ENCODE that grow much faster. So that's a measure of success of a project like this at the community level. A latest very large project, I'm not personally involved, but Roderick Guigo, the coordinator of our program, is a major player in that one, is GTEx. So GTEx was, you know, anytime you go to anytime you have your annual checkup, and I guess most of us roughly the same age, you get your medical checkup, and every year you see yourself decaying because your numbers are moving to the left or to the right of the accepted ranges of value, you know, cholesterol, whatever you name. Yeah, you know, I used to be very proud to be right in the center, and you really see yourself decaying. You have no clue, nobody in this room has any clue what is a normal level of gene expression in your tissues. We don't know. How should your RNA polymerase be expressed at which level in a healthy liver, population-wide? This is something that is totally unknown to us. And that's exactly what the GTEx project was about. Trying to characterize the level of transcriptomic activity in individuals across the population. So it's a very, very big project where in the US, they try to enlist as many, through the family obviously, many recently deceased people to have their key organs deep sequenced in order to establish this reference catalog of human variation at what we consider to be the lowest phenotypic level, the molecular phenotypic level, which is the RNA. Okay? And so it's one of these military consortium, because if you're talking about sequencing entire individuals, This is really complicated. This goes down to, in some of the work packages are dedicated to engaging the families. You have psychologists and all these kind of things, because time is the essence. If you want to do good RNA, you have to do it very early. The main confounding effect on this analysis is the time between death and sequencing. Okay, so it's a very, very sophisticated project that has been published. There was almost an entire issue of Science dedicated to this thing. And this is only starting. I mean, the data is here, but it has only been very, very superficially analyzed. And it's very clear that all these AOPs and all these things you guys are very interested in will be very, very interestingly impacted by this type of data. Okay, now moving into how much time do I have? 2 minutes then. I'll try to be fast, sorry. So metadata is very important, as you know, the way we describe data. And that has been a huge effort in the ENCODE consortium to come out, and they were kind of pioneering these things. So there was a strong interaction with the EBI, figuring out the right ontology and all these sort of things. Because you know that if you do not describe the data right, you'll be unable to use it. The type of databases that were static objects where you would go and pick up one word like you would pick up a word in a dictionary is finished. Now you want really data to be interoperable across the entire database so that you can ask very sophisticated questions correlating one factor with another. And we are still very far from this. But ENCODE has been a very important step forward. Here is a very nice example of why metadata is useful in a kind of boring way. Here we are looking at interaction between genes. And if this was a student coming in your lab with this graph, you'd be very happy. You have a strong, very nice effect. But then if you start superposing metadata, well, you'd like the cell type to be telling you something. They don't seem to cluster very much. And the only thing that actually clusters very much with this very strong signal is actually the protocol that was being used. So what you're looking at here is a batch effect. And batch effects, some are very easy, some are very, very complex. Metadata is the only way to make sure that batch effect is not a confounding factor. Another aspect I want to insist, which is an issue, most people take for granted that computers that wet lab is complicated, you need to replicate. Computer analyses are stable, you don't need to replicate. This is true for simple analysis. This is not true. This is not true for much more complex analysis, like the kind of omics analysis we do. I'll show you here a very simple example. This is something we do a lot in my lab. This is phylogenetic tree reconstruction. Okay, you take a bunch of sequences and you compute a tree. We use these things, the length of the branches, to make a lot of functional inferences. Now, it's a fact that if you use the same input data, exactly the same software, same version and all these kind of things, and run this on the Amazon Elastic Cloud, on your own cluster, on a Mac, you will get slightly different results. And this can have a major impact. If you're doing this on 1 million of different assays, different proteins, this becomes a hidden bias. So we work a lot on trying to make sure that these outputs become stable. And for this, we have developed tools. For instance, Nextflow is one of them that uses something that is called a container technology. And I'm going to skip this. It's not so important. But the idea is to be able to run all the kind of analysis. Most of the analyses done on GTEx and ENCODE are difficult to reproduce. You run them one day, you run them 6 months later, it's difficult to get exactly the same results. And one of our objectives is to make sure that this problem goes away. The irreproducibility of computational analysis should not be a confounding factor, which it currently is. Okay, so these are people using these kind of things, and then so we are very happy to have a base of users growing very fast. I'm done. I want to acknowledge people working on this thing, especially Roderick Guigo, who has been a major PI in ENCODE and GTEx, Arkady Navarro, who heads the EGA, an Elixir-based resource for the storage of genomics and phenomic data related to human health. And Paolo Di Tommaso, who developed Nextflow. Thank you for your attention. Sorry for